Good day everyone. Today we will be going to summarize on the video how bacteria talk. How can bacteria do something together in a community? Bacteria talk to each other with a chemical language. They secrete small molecules that are hormones. Let's take Vibrio fischeri as an example, where a lone bacteria will not light up, while many bacteria will create bioluminescence. When bacteria multiplies and are all making these molecules, the extracellular amount of the molecules increases in proportion to cell number. The molecules hit a certain amount that tells the bacteria how many neighbors there are. Hence, they recognize that molecule and all the bacteria turn on light in synchrony.
So, why do bacteria do things in communities? Living in a community allows for quorum sensing, a type of chemical signaling to occur within a bacterial population. This allows it to control the pathogenicity. As an example, 10 or 20 scraggler bacterium cannot efficiently take down a large host such as a human. However, through quorum sensing within the community, which allows for the counting of cell numbers, they're able to tell when the community is large enough to launch an effective attack against the host. In other words, it provides strength in numbers. The mortality rate of that disease goes up due to the increased virulence, and so does the longevity of the bacterial species. The term pathogenicity is the ability of a bacteria to cause disease and harm the host organism. Meanwhile, virulence is the extent of pathogenicity and is typically correlated with the ability of a bacteria to multiply within a host.
Mechanism of bacterial quorum sensing. For the bacteria to use quorum sensing constitutively, they must possess characteristics. 1. To secrete a signaling molecule. 2. An autoinducer to detect the change in concentration of signaling molecules. 3. And to regulate gene transcription as a response. Gram-positive bacteria and gram-negative bacteria differ from the mechanisms they use in quorum sensing.
Moving on to parallel interspecies signaling. It refers to the sending and receiving of chemical signals between different bacterial species, and autoinducers are responsible for the exchange of chemical signals between the different bacterial species. Autoinducers are molecules that are produced with regards to the change in cell population density, and these autoinducers allow gram-negative and gram-positive bacteria to communicate with each other for physiological changes.
New antibiotics that target quorum sensing of bacteria. Bacteria relies on quorum sensing to synchronize many activities, including the production of virulence factors and the formation of biofilms. A new field of drug development targets good beneficial traits that offer little selective advantage to resistant bacteria. As quorum sensing controls good behaviors, it has become a prominent target for new antimicrobial drugs. Adding the bacteria's own signaling molecules to a sample makes cells think that more neighbors are present. To stop bacteria from sensing their neighbors, we can mutate the receptors of bacteria that bind the molecules. Thank you for watching and have a great day ahead.